Alright, so I did my presentation on cloning of adult animals. This is an overview of SCNT, which is somatic cell nuclear transfer, and it's used to clone adult farm animals. Alright, so in 1997, scientists announced the birth of Dolly, which is the first ever cloned adult animal. To this date, sheep, goats, cattle, mice, pigs, and other animals have been cloned using somatic cell nuclear transfer. The challenge of cloning with this process is reprogramming the somatic cell of the nucleus to develop the embryo. Genetic modification of farm animals provides a means of studying genes involved in a variety of biological systems and disease processes. Moreover, genetically modified farm animals have created a new form of farming, whereby farm animals serve as bioreactors for production of pharmaceuticals or organ donors. A major limitation of cloning procedures is existence for producing live offspring. Dolly was the only live offspring produced after 277 attempts. The ability to clone mammals by simple nuclear transfusion was thought to be biologically impossible. Furthermore, the demonstration of totipotency of mammary nuclei inferred that other types of somatic cells can be readily obtained and cultured in the laboratory. The procedures of somatic cell nuclear transfer immediately help promise offering an alternate way of introducing targeted additions or deletions of the genome of farm animals. And so the totipotency is the ability of a single cell to divide and produce all the different cells in the organism, which brings along the extra embryonic tissues. And somatic cell nuclear transfusion is a laboratory technique for creating a clone embryo with a donor nucleus.
This video was prepared here by my colleagues Dieter Egli and Kevin Hagen. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. If we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side. It is held by this holding pipette on the left. They're drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. Pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again. A little hole in the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.
Alright, so now is an example of egg being enucleated, which takes out the nucleus of that cell and puts in a different one from a completely different somatic cell, and that's the process of somatic cell nuclear transfer. Dye was not the result of a union of the oocyte and sperm, but derived from nuclear gelmine contained with a latent induced mammary cell, and the cytoplasm originated from a nucleated oocyte and somatic cell. That's what happened in that video right there. The process is often complex, and it's remarkable that it even happened during this cloning. Very little is still known about the factors of somatic cell transfers, and the ooplasm is the cytoplasm of the egg or the ovum. This current lack of understanding continues to reflect of the inefficiency. So Dolly was the first clone of an adult mammal after 277 attempts. So it's pretty low. And the focus of this overview is to provide information about the SCNT and procedures used to clone animals. So here are the methods and procedures. The ultimate challenge of cloning using somatic cell nuclear transfer involves reprogramming of somatic nucleus in a manner conducive with embryonic development. Predominant cell type choice for reprogramming somatic nucleus is an oocyte arrested at metaphase 2. Matured oocyte is a logical choice as critical components require its initiative in directing the early embryo development, often contained in the oocyte. The first step of cloning procedure involves a collection of somatic cells from the animals to be cloned. Choosing somatic cells often vary. There's a lot of cells you can choose from, such as the cumulus, fibroblasts, mammary, the muscle, or the uterine cell, which may work better than other cells to produce an offspring. After collecting somatic cells, they may be utilized immediately after the long-term culturing when it's put together. The second part of this is the hardness, which is when we use SCNT, it requires removal of the material DNA from the oocyte arrested at metaphase 2. Doing so requires microtumors, which we saw in the video. The next step of SCNT is to insert the somatic cell into the egg cytoplasm. This constructs the equivalent of one cell embryo. In most farm animals species, is performed with electrically induced, which kind of gives it a shot, which basically acts like fertilization. Initially, a somatic cell is mechanically inserted into the perivitelline space using microtools. After that, the resulting couplet of the egg and the cytoplasm and the somatic cell is aligned between two electrodes and pulsed with electrical current, which acts as fertilization. Electrofusion is important on maintaining contact of somatic cells with egg cytoplasm so that the membranes of each cytoplasm may intermingle with each other. The different applications of SCNT, there's a number of them, and have been described as benefiting animal agriculture and human medicine. Success in cloning of dog, sheep, cattle, pigs, and goats confirms the usefulness of SCNT for the purpose of colonial expansion of agricultural important food producing animals. So using SCNT in cloning animals allows more livestock to be produced in a way. There's also some dangers to it. They're still investigating it, and that's why a lot of it is still unknown. It's really new procedure. There's also an increasing interest to utilize SCNT to restore endangered or extinct species. SCNT is rapidly becoming the method of choice for producing transgenic farm animals. Moreover, the use of SCNT increases efficiency of producing transgenic farm animals. to 100%. Production of transgenic farm animals provides one with the means agricultural and biomedical disciplines for studying importance of genes involved in a variety of biological systems. Transgenic animals are used as bioreactors for production of pharmaceuticals or even organ donors for human populations, which I hope to use in the future with SCT. And the announcement of the first clone sheep, Dolly, was like a very promising way for them to use this for humans in the future for possible organ donation. And yeah, and they're going to use other animal cells to help that with pharmaceuticals and whatnot. And here's the resources. It's just this is just a picture of the sheep Dolly that was cloned. That's it.